Welcome to section 7.4, where we need to go over the concept of cellular transport. So with cellular transport, there's a lot of materials that we don't just need to get around inside the cell. We've got vesicles for that, we've got ER for that, we've got the Golgi apparatus to help out with that, but we also have to get items into and out of the cell itself. Now, for many things, we can use passive transports. This is going to be one of our first big ideas in transport. Now, passive transport is called passive because it requires no energy. So specifically, this is not going to need ATP, that energy molecule that we've been bringing up from time to time. The other idea is this is going to involve molecules moving from where there's a higher concentration to where there's a lower concentration. So like if you look at the picture here off to the side, you'll see in this first part, we've got a high concentration of blue on the left-hand side. We've got a high concentration of red on the right-hand side. Now, if we remove this barrier, the blue molecules, as they bounce around randomly, should end up spreading out naturally. So this isn't something that they necessarily think about. It's just something that happens energetically because they're moving. They tend to bounce around and spread out because of that. So the blue particles are going to randomly start to move over towards the right, and the red particles are going to start to randomly bounce around towards the left until eventually we reach this equilibrium. We reach this point where they're balanced out. And so now they're both evenly spread throughout the container. This happens with just about anything that you have that's a gas. This will also happen in liquids. You can put food coloring in water and watch it disperse until it's evenly spread. You can take and spray cologne or some scent in the air, and it will ultimately diffuse through the air until it evenly covers the room. Now we're going to focus on this happening not just in a room, but across a cell membrane. And there's going to be three basic categories that we're going to discuss. So diffusion will be our first, then we'll move to facilitated diffusion, and we're going to wrap up passive transport by talking about osmosis. So these will be the three types of passive transport you need to know. So with the first type, and really this is the one that's the main type, because the other ones we talk about will be kind of subtypes of this. They'll still be diffusion, they'll just be special cases of diffusion. So the first part we talk about, we're just going to call diffusion, but you might also see them say it is simple diffusion. And that just means it's going through the actual phospholipid bilayer. It's not going through with any help. There's nothing fancy going on here. And so this works for particles that are able to pass through the phospholipid bilayer. So once again, this is going to work with things that are small and polar or are relatively small, small to medium size and nonpolar. These guys will be able to pass through these phospholipids, and they will do so down the concentration gradient. So what I mean by that is they're going to go from high concentration, that's up at the top here. At the bottom you can see there's not many of them. In this case it appears none at all. And so they're going to want to move from high to low or down the concentration gradient. It's just another way of saying the same thing. And because they, by just randomly kind of bouncing around, will spread out like this, they will go down the concentration gradient without any help. This will once again not require energy. That's why it's passive. Now we can speed this up if we want. If we raise the temperature, because this is due to them bouncing around and moving, raising the temperature makes them bounce around more, it makes them move faster. So it would make diffusion occur faster. We could also increase the pressure, which is going to, yet again, help them move around faster because they're going to collide more. The more collisions they have, the faster they can spread around. So there's a variety of ways that we can try to up the amount of collisions, we can try to up the speed of the particles, and this will help us increase the rate of diffusion that happens. Now, once everything is set at the end, we see that they have reached this state of equilibrium, which we're going to call dynamic equilibrium. The only reason it gets this fancy dynamic word is because dynamic implies there's still movement, and there is. When you look at this, you'll see there's about equal numbers of particles on both sides, but that doesn't mean they stay there. You're going to see some of these particles will go across, in this case down, and you'll see some of the particles from below will move up, but they'll do so in about equal amounts. And so you might see them discuss the fact that at dynamic equilibrium, there's no net movement. It's not that there's no movement. There's always movement. These particles are moving. But ultimately, if one guy kind of bounces up, you should have another guy that randomly bounces down. It's kind of like having a room where if one person leaves, somebody else enters, so overall, the population of people inside the room stays the same. Now, Now, facilitated diffusion is going to be a fancy case of diffusion. So this is going to deal with these ions that have this charge, which we've talked about. The middle part of the phospholipid bilayer does not like because that part is nonpolar. This will also work with things that are polar that are a little bit larger. Really small polar guys like water can sneak through, but if you're any larger than that, it's too difficult being partially charged and trying to get through this nonpolar tail region of the phospholipid bilayer. So these guys can't get through the phospholipid bilayer directly, so they've got to get help. And that help comes in the form of a membrane protein, or you can call them transport proteins, or you might see them called carrier or channel proteins. It's similar idea. It's some protein that goes through the membrane. And depending on the exact type, some of them just have like a tube, so they look more like this middle guy. So ions can just kind of shoot through. Those are called channel proteins. And some of them, like the one it's demonstrating here, are called carrier proteins because the guy in question will bind to it. That'll cause a conformation or shape change, which then allows that substance that bound to be released and go to the other side. So I always like to think of this as a plastic bag, where if you've got where you're walking your dog and your dog happens to do his business, then ultimately you might not want to touch it. But all you've got is a plastic bag, no shovel. So what you can do is invert the bag, grab the business, and then flip it inside out. And so now what was outside the bag, by flipping it, just became inside the bag. So it's very similar where a carrier protein will change shape, and so what was outside the cell is now inside the cell. You know, if it binds here and this is the outside, when it changes shape, it can release it inside. And so this is one of the ways we can get some of these molecules that cannot go through simple diffusion into the cell. We just have to have the proper membrane protein to allow this. So keep in mind, proteins that are in the membrane only work with specific molecules. So they'll work with like one ion, or they'll work with one polar molecule. So don't expect that anything that's ionic or anything that's polar can come in through a membrane. They are limited in terms of who they let in, based upon which proteins are present in the membrane. They're also limited where if something gets to be too big, they can't fit through a membrane. You know, even membrane proteins have a size limit with what can fit through them. So things that are really big would not be able to use this avenue. But otherwise, it helps us out.